Hello dear learners. In this video we have to talk about the another types of mutations which can occur in the cell. That is when the mutations occur in relation to the changes in the numbers of chromosomes. So when the numbers of chromosomes get altered, these kind of mutations are related to the quantity of the chromosome mutation. So they can be of two types, one of them being the aneuploidy and the another one of them being the polyploidy. In the aneuploidy, either the addition or deletion of 1, 2 or 3 chromosomes can occur. But in the polyploidy, entire set of chromosomes get duplicated or triplicated. Let us take an example. In our gametes, in the haploid condition, they have 23 chromosomes. Now, if the entire set of 23 chromosomes get duplicated in the gamete, that time we would have total of 46 chromosomes. This condition is referred to as polyploidy. But in the another condition of aneuploidy, only one or two of the chromosomes can be added or can be removed. Now in the aneuploidy, there are various reasons because of which the aneuploidy can occur. One of them being due to the centromere deletion. So what happens is, we know that the chromosomes consist of this centromere. Through this centromere, the spindle fibers pull them and then the anaphase will occur. And these chromosomes will segregate into the progeny cell. Suppose if the spindle fiber is absent, in that condition these chromosomes won't be able to segregate and this will create a problem. Let me change the color. So this can create a definite problem. Then the another kind of cause can be Robertsonian translocation. Robertsonian translocation. This we were discussing in our previous video. In the Robertsonian translocation, which generally occurs in the acrocentric chromosomes, wherein the centromere is present to the very terminal. After the crossing of one of the chromosome consists of two very long arms and one chromosome consists of very short arms. This chromosome, which consists of very short arms, this is eventually lost in the cytoplasm. And that time we will have this removed and the condition will arise of aneuploidy. The third cause can be due to the improper orientation or the non-disjunction of the chromosomes. So what basically happens is, during the metaphase, the chromosomes have to align on the metaphase plate, and after that in the anaphase they have to be separated. But if the chromosomes do not align properly, that time we say the condition of non-disjunction has occurred, and that time the aneuploidy can be possible. Now there are various kinds of aneuploidies which we have to discuss here. These aneuploidies are nullisomy, monosomy, trisomy and tetrasomy. In nullisomy, what happens is, one complete pair of chromosomes is removed. Suppose in an individual, the chromosome 2 consists of two copies because the individual is diploid. Now if this complete two chromosomes are removed, that time the condition arises of nullisomy. And the peculiar representation of nullisomy is 2n minus 2. So originally we should have 46 chromosomes, but then we only have the 44 chromosomes. In monosomy, only one of the chromosome is removed, and here we have the 45 chromosomes. In trisomy, rather than removal, one of the chromosome gets added. So there is a Down syndrome. In Down syndrome also there is a trisomy of chromosome 21. So there are 3 copies of chromosome 21 which are present, and this is the classic example of translocation as I told you. The next case is tetrasomy which is 2n plus 2. So these are the various things which can occur, or which various kind of aneuploidies which can occur. Apart from this, there are another kind of aneuploidies also. For example, I had the examples here. Nevertheless, I'll tell you like this way. One of them being double trisomic. Now double trisomic is when two different chromosomes have the trisomy. So it is plus 1 plus 1. Please do not confuse it with tetrasomy. It is different from tetrasomy. Similarly, we can have double monosomic. Double monosomic. In this condition, there are two different chromosomes whose 1 copy each is lost. So it is 2n minus 1 and minus 1. And then similarly, we can have double tetrasomic also. But it is very, very rare. So these are the various types of aneuploidies which can occur. So basically in the aneuploidy also there is the gene dosage which is altered but not the DNA sequence, because either the chromosome will be absent or will be present. So here the genes will either be absent and present, but the DNA sequence itself is not altered. The whole of the DNA sequence either can be deleted or can be added, but not individual mutations can occur on the DNA sequence. So that is very important to know. Now we have to talk about the another form, that is the polyploidy. So here we have the polyploidy. As I was talking about, in the polyploidy, the entire set of chromosomes can become duplicated. When they become duplicated, it is 2n. When they become triplicated, it is 3n, and it can even be reaching up to 4n. These conditions are very normal in the plants, but in animals, they are rare. Basically, in the lower invertebrates, we can see them, and even in some of the frogs and salamanders, and one of the rats which live in Argentina. In that, we have seen the condition of polyploidy. Now, polyploidy can itself be of many types, one of them being the autopolyploidy, and the another one of them being the allopolyploidy. Now, let us try to discuss them in detail, that what is the meaning of autopolyploidy and what is the meaning of allopolyploidy. Autopolyploidy generally occurs in the or when the chromosome sets they come from the same species. So that time the autopolyploidy can occur. Now this is due to the non-disjunction or due to no cell division. So suppose here, if we are talking about the autopolyploidy through mitosis. So in mitosis, so generally only one organism or one individual is required, even not two individuals. So in that condition, in that particular individual in its own cell, suppose the non-disjunction has occurred and the division has not occurred. That time this has to be converted to two cells, but it did not. So now it will be having, rather than becoming 2n, it will become 4n. And if we talk about meiosis, in meiosis also what happens generally, we have two members involved here, their gametes are involved but they are from the same species, because we are talking about autopolyploidy. So here also in this way it can happen, that what has happened is that there is one individual, in his or her gametes the meiosis did not occur, because of which his or her gametes became diploid, and the another individual was normal who had the haploid gametes. In that condition, when the two of them fused, the resultant zygote became triploid. So this is the condition of polyploidy, but the polyploidy, which is within the same species. Now there can even be the allopolyploidy. Allopolyploidy means there is the polyploidy condition because of the different organisms from the different species. So let me take the example here. Let me try to conclude it. See, I'll try to explain here only. So see, what happens is the allopolyploidy. This is again a kind of polyploidy, but it is occurring by two individuals. Means let's say this is individual 1 and this is individual 2. This is producing a gamete. This is also producing a gamete, and the two of them are fusing, or they are made to fuse artificially. So this is belonging to species 1 and this is belonging to species 2. So that time the allopolyploidy can occur. Now suppose the gamete here was 2n and here also it was 2n. So this became 4n. Now this is a difficult situation because the two species were different and their chromosomes were also different. So in the cell also, in the gamete also, it was in this phase only. Not, they did not fuse actually. They were in 2n plus 2n condition. Now sometimes what happens due to rare mitotic disjunction, what happens is this particular pair, it is capable to undergo meiosis and it can undergo normal meiosis, and this particular set of chromosomes is also capable to undergo normal meiosis. Now if you try to analyze this whole, it is tetraploid. And what kind of tetraploid? It is allotetraploid. But if the two particular sets, they can undergo normal meiosis and then they can produce the normal viable and the fertile gametes, that time this allotetraploid can or be also called as amphidiploid. So this is the whole concept of alloploidy. So here we also have covered the numeric variations which can occur in chromosomes and which can lead to the mutations.